Hello everyone and welcome to another video on Nowhere Udaris. This is your host Mahmoud Nowhere. Today we'll be going through biochemistry's own glycolysis and carbohydrate metabolism. Alright, I don't know about y'all, but glycolysis and all the cycles associated with it to produce ATP and NADH and stuff like that, they have been difficult for me to memorize. But today we're gonna develop, and I'm gonna tell you a way to kind of simplify the matters and kind of understand it more. So as always, please leave any comments and like, share, and subscribe. And shall we get started? So we're gonna be speaking about glucose transporters, but today we're gonna focus only on two. There are four glucose transporters. There's GLUT1, GLUT2, GLUT3, and GLUT4. Alright, but today we're gonna only focus on GLUT2 and GLUT4. So GLUT2 versus GLUT4. GLUT2 is a low affinity transporter in pancreatic cells, and it has a high substrate concentration. It's usually 15 mm, which is higher than the normal blood's concentration, and absorbs excess glucose for storage. So when the glucose concentration drops below the concentration substrate for the transporter, excess bypasses liver and enters the peripheral circulation. And then because the Km, the substrate concentration for GLUT2 is too high, so when blood glucose is high, liver stores the excess glucose as glycogen, which we'll be talking about later. It also serves as a glucose sensor, alright, along with the glucokinase. They both serve as glucose sensors for insulin release. And then next we have GLUT4. GLUT4 is kind of what we're gonna be focusing on a little bit, in which it's found in adipose fat tissue and responds to glucose concentration in peripheral blood. The rate of the glucose transport for these tissues increases when insulin increases, which increases the GLUT4 production, because you see, insulin is released when there is a high blood sugar, when there is like a high concentration of sugar in the blood. Okay, so insulin is released in order to lower that. So because GLUT4 is in fat tissue and kind of transports the glucose into cells, insulin kind of triggers more of its production in order to lower the blood sugar levels. Okay, so like we said, GLUT2 and glucokinase are glucose sensors for insulin release. So GLUT2 also serves as glucose sensor when the concentration in the blood gets too high. GLUT2 and glucokinase, they're like, hey, insulin, okay, get released by pancreas beta cells. They release that stuff. Alright, and then in the muscle, when you have excess glucose, that glucose turns into glycogen, and this is stored glucose in the liver. So in the liver and muscle, excess glucose is turned into glycogen to be stored and used and broken down later. We're going to be learning about how we make glycogen and how we break it down. And then in adipocytes, which is adipose fat tissues, excess glucose actually turns into DHAP and then turns into glycerol phosphate. Glycolysis converts glucose. So now we're going to go into the deep stuff. Glycolysis converts glucose, which is a 6C kind of molecule, into two pyruvates, 3C and 3C, and it converts it into NADH. So glycolysis can kind of transport the NADH to be fed into aerobic respiration to generate energy. So glycolysis occurs in the cytosol, alright? And then, like I said, the products of glucose glycolysis breakdown is basically ATP, NADH, and two pyruvate molecules. Okay, NADH and pyruvate and ATP are used in later processes such as Krebs cell and stuff like that, and etc. Alright, we're gonna just break down some of these. So hexokinase, glucokinase, and phosphofructokinase and pyruvate kinase, these are irreversible stages or irreversible enzymes, and these enzymes help with the breakdown from one step to another. So hexokinase is present in most tissues and has low concentration. That's why kind of in all the processes we're gonna see glucose using hexokinase to go into the next stage. Glucokinase, like I said, acts as glucose sensory and has high Km, has high substration, same as GLUT2. GLUT2 and glucokinase have high substrate concentration. And then phosphofructokinase inhibited by ATP but activated by AMP. So high ATP turns off the cycle and high AMP turns it on. Um, the only reason we have like kind of glycolysis in order to generate ATP and generate energy and pyruvate for respiration, but when we have too much ATP, you don't really need or have a need for the cycle that much, right? And phosphofructokinase 2 is kind of controlled by insulin, which we'll discuss later. And glycerolaldehyde 3-phosphate dehydrogenase is responsible for the oxidation and the addition of inorganic phosphorus to glycerolaldehyde 3-phosphate, and it reduces NAD plus to NADH as a byproduct of glycolysis. Alright, so let's go through glycolysis, okay? Focus with me, please. We're going to go through the process of glycolysis. Alright, just focus on the right side more of my screen and leave the left side. We're going to come back to it, okay? So, for glycolysis, you always start with glucose, okay? And then you use hexokinase, okay, to turn glucose into glucose 6-phosphate, okay? And I kind of developed, because this is going to be too big for an acronym, so I kind of developed shortcuts, okay? So the first shortcut I call GH, and it kind of reminds me of like growth hormone, okay? GH, the first part of the acronym, first letter, is for what's being broken down. Then the second part is for the enzyme it uses. So GH, glucose, goes into glucose phosphate and it uses hexokinase, so GH. And then we have G6P. And then G6P uses PI, right? So G6P, glucose 6-phosphate, uses phosphoglucoisomerase, my bad. And then G6P wants to go into fructose and then uses fructose, goes to fructose using phosphoisomerase. So this acts on this and then this acts on this, okay? And it kind of helps you predict the next breakdown, and then goes into fructose, okay, and then fructose 6. Alright, and then fructose 6, it's not here, but fructose 6 goes into, it uses PFK2, so here it's called PFK. So we have two fructose 6-phosphates, okay? So fructose 6-phosphate goes into fructose 6 biphosphate using PFK2. So biphosphate, FB, you can see that as FB, and then FB, fructose 6 biphosphate, uses PFK1. Alright, we're gonna speak about PFK2 again, okay? And then the phosphofructobiphosphase goes and becomes fructose 1,6 biphosphate. So we can just say F16B, and then this F16B uses aldolase to go into to go into G3P. So far we have the first half, okay, GH, and then we got G6P, and then G6P uses phosphoglucoisomerase PI to go into fructose, and then fructose uses PFK2 to go into fructose biphosphate, fructose 6 biphosphate, and then fructose 6 biphosphate uses PFK1 to go into fructose 1,6 biphosphate, and then fructose 1,6 biphosphate goes into what we call G3P using aldolase. Alright, so we have G3P, easy number. Okay, so G3P uses G3PD, so glyceraldehyde 3 dehydrogen kinase, something like that. Dehydrogenase, my bad. Alright, so uses G3PD. You can just add a D if you want. I found that to be helpful. Just as adding a D. I should just change the font. Okay. I think that would be more helpful if I change the font. I mean the font color, my bad. And then this is PFK1, and then this is PFK2. So 2 and 1, 2 comes before the 1, and then here PI. And then PI. Okay, and then after G3PD, it needs to turn into what we call 1,3 biphosphate. So it can be 1,3 BP using the dehydrogenase, the G3P dehydrogenase, and then 1,3 BP turns into 3 phosphoglycerate using something called pH. I just call it pH for short, which is phosphoglycerokinase. Alright, so pH, remember, like pH, acidity, and stuff like that. Okay, and then now it comes the easy part. Okay, trust me, this is the easy part. So 3 phosphoglycerate, I call it 3 PG, point guard if you're into basketball, and then 2 PG. Okay, so here is just gonna be mutase and enolase. Alright, mutase and enolase, and then we go from 2 phosphoglycerate into PEP, something called PEP. So PEP, and PEP uses pyruvate kinase. Kind of easy to memorize. So PK, so you can kind of remember this 1,3 biphosphate phosphoglycerate pH, and PEP is PK, kind of close to pKa, so kind of in the realm of like chemistry and stuff like that. Alright, and then uses PK pyruvate kinase to go into a pyruvate, which is the final final product that we want. Alright, so let's review. G glucose uses hexokinase to go into G6 glucose 6 phosphate, and then glucose 6 phosphate uses PI phosphoisomerase to go into fructose 6 phosphate, and fructose 6 phosphate uses PK2 phosphofructokinase to go into fructose 6 biophosphate. Alright, and fructose 6 biophosphate uses PK1 to go into F16 biophosphate, and then F16 biophosphate uses aldolase to go into G3P, which is the glyceraldehyde 3 phosphate, and then this uses G3P dehydrogenase to go into 1,3 biphosphate glycerate. Right, and then it uses pH phosphoglycerokinase, because you see I'm using these kind of short forms because it's going to be way harder for you to memorize the full name, so just memorize these. And then uses this to go into 3PG, and then uses mutase to go into 2PG, and then 2PG uses enolase to go into PEP, and PEP uses pyruvate kinase to go into pyruvate. Alright, that was a lot of effort. Anyways, so 3 phosphoglycerate or 3PG kinase transfers high energy phosphate from one BP to ADP. So you see, it kind of transfers 2 ADP into 2 ATP, or ADP into 2 ATP, as going from 3 BP into 3 PG. And then pyruvate kinase, which is the last enzyme before you make pyruvate, feed forwards, which means stimulates the activation of the enzyme downstream, activated by 1B biophosphate from the PFK reaction. So this whole thing is called a feed forward activation. So hexokinase acts on this to activate this, and this acts on PFK to activate the PFK1, and then PFK1 acts on this, and this, and this, and this, and that, and they all act on each other in order to trigger pyruvate kinase, which triggers a pyruvate. And then the total net is 2 ADP, alright, as we see here, being produced, plus 2 ATP plus 2 NAD plus glucose is equal to 2 ATP and 2 NADH plus 2 pyruvates. What if we don't have oxygen? So in the absence of oxygen, pyruvate ferment to regenerate NAD plus. Okay, and next I was talking about PK or PFK 2, and how insulin increases PFK2 in order to increase the fructose 1,6-biophosphate, which increases PFK1. So insulin is a really strong player in glycolysis because insulin wants to break down the glucose as fast as possible, wants to lower the glucose in the bloodstream. Okay, so it wants it to start its glycolysis, so the glucose can be turned into pyruvate and can kind of lower the blood sugar, the concentration of blood, the concentration of sugar in the blood, and turns it into something else. So that's why insulin kind of increases and doesn't inhibit this process. Glycogen on the other hand decreases PFK, which decreases PFK 1. Glucogen on the other hand tries to inhibit PFK2 from doing this, because glucogen wants to be broken down and wants to be used in order to increase the levels of sugar in the blood. Okay, in the presence of O2 pyruvate, using pyruvate dehydrogenase, NAD plus converts into NADH plus CO2 plus acetyl enzyme, which we'll be talking about. So after pyruvate is produced, we have 3 outcomes for this pyruvate. So the pyruvate can turn or convert into acetyl CoA by enzyme called pyruvate dehydrogenase, or can convert into lactate, or can convert into oxaloacetate. So it can convert into lactate by lactate dehydrogenase, and it can convert into oxaloacetate by pyruvate carboxylase. Right? Glycogen breaks in the liver to maintain constant glucose levels in the blood. Okay, glycogen wants to maintain glucose level, and glycolysis wants to break down glucose into pyruvate for cellular respiration. And muscle glycogen breakdown to provide glucose to muscle during vigorous exercise. Next, we have glycogenesis. Okay, so focus on the wording here, because wording really matters, because maybe you get a question about the difference between glycogenesis versus glucogenesis versus glycolysis versus glycogenolysis. Right, so these are different processes. We just went through glycolysis conversion of glucose into pyruvate. Alright, next we have glycogenesis. Glycogenesis is the synthesis of glycogen in the cytosol. Okay, so glycogenesis is a process in which glucose is converted into glycogen for storage in the liver and muscle cells. So the way this process happens is we're also going to start with GH. Oh, I forgot to say something. With the first process, glucose into glucose 6-phosphate, it's hexokinase and glucokinase in the liver. So during glycogenesis, because it kind of happens to be stored in the liver, glucokinase is here. So here it's not just going to be GH, it's going to be GHG. So I don't know, pretend it's a label or something. GHG. And then GHG converts into GPH6. So let's do this again. G, and then you got HG. Okay? And then it converts into G6P. Right? So these act on the glucose in order to convert it into glucose 6-phosphate, and then glucose 6-phosphate uses PM, phosphoglucomutase, right? So it uses mutase. You remember we kind of used mutase at the end. Here we're using it early on to convert into G1P, sorry, G1P, not G16. Alright, and then glucose 1-phosphate uses UDP glucose. Glucose phosphorate. So uses that. You can just say UDP. I don't have space here, I don't know why. And then converts it into UDP. Okay. So UDP glucose added to growing alpha 1,4 glycogen chain. Alright. And then UDP uses glycogen synthase, and glycogen synthase is active when blood glucose level is high, alright, and deactivated when sugar is needed. Okay, so glycogen synthase is activated when blood glucose levels are high. You need to bring them down, alright, and then you bring them down by increasing insulin. Okay, insulin brings that stuff down, and deactivated when sugar is needed. Okay, so deactivated when sugar is needed because glycogen is going to be broken down, but right now glycogen is being made, so that's why glycogen synthase is being activated. Okay, we're going to just call it GS. Okay, GS. I know it's a lot to take in, but kind of this is the best way to memorize them, unless you can memorize the whole word. And then UDP turns into or adds on to the alpha 1,4 glycogen chain. Okay, alpha 4 glycogen chain, and then the alpha 1,4 glycogen chain uses branching enzymes. So you can just call these BE, branching enzymes, to turn into a glycogen. So the final product is a glycogen. So glycogen synthase introduces the alpha 1,4 bond to glycogen chain, and branching enzyme hydrolyzes a alpha 1,4 bind and moves them to make a branch, hence why it's called a branching enzyme. And then glycogenin is a core protein that a glycogen uses at its core, helps in converting glucose into glycogen. Alright, so glucose to glucose 6-phosphate, and then glucose 6-phosphate to glucose 1-phosphate. So far we have three glucose. It goes glucose to glucose 6 to glucose 1, alright, and then from glucose 1 it goes to UDP, and then from UDP it goes to the alpha 1,4 glucosyl units, alright. They make the units so they can be branched later and make a glycogen. They add up, they make a glycogen, okay. This glycogenesis is way simpler than the glycolysis for sure. Next we have glycogenolysis. Alright, kind of remember it. Don't, like, for me, when I remember stuff, remember it by the ending. Genolysis. Alright, this is glycogenesis. No L. This one has an L. So the one with an L is basically the breakdown of glycogen into glucose. Alright, now the body needs glucose. Where is it gonna get the glucose from? It's gonna get the glucose from the stored glycogen in the liver and the muscle. And since Ramadan ended, this is how we were all surviving Ramadan, because we have stored glucose in the form of glycogen in the liver and the muscle, and then glycogenolysis happens, where like the body kind of, you know, uses the resources it has, and then when it runs out, it converts glycogen into glucose. All right, using glycogen phosphorylase and debranching enzymes to make glycogen, but debranching enzymes is to break down glycogen. Alright, occurs primarily in the liver and the skeletal muscle when the body needs glucose for energy. And then we have the last process, glucogenesis. Alright, glucogenesis is the process of glucose synthesis from non-carbohydrate sources such as amino acid, lactate, or glycerol. I want you to think of glucogenesis as like upside down glycolysis. Alright, so it starts from the pyruvate kinase and makes its way to glucose. Alright, it's not literally an opposite of glycolysis, but that's the easy way to just think about it. And as always, if you have any questions, let me know, and as always, have a blessed